Hello everyone, welcome back to Biomix Hub. In this video, we are going to discuss about the question of nucleic acid which have been asked in previous year Life Science Net. So, we will discuss only two questions of the same topic. So, this is our first question and this is the second question. So, before discussing this question, we need to be clear about the nucleic acid. Okay, the nucleic acids are the purines and the pyrimidines. First, we will discuss about the purines. Here we will discuss about purine. When we talk about the purines, it is having double ring structure. This is the structure of purine. And the numbering will be started from this position 1, 2, 3, 4, 5, 6, 7, 8 and 9. And these are made up of nitrogen and carbon atoms. And it will be in the sequence NCN, CCC, NCN. This is a sequence. So, in the first position, the N will be present. In the second position, carbon will be present. In the third position, nitrogen will be present. In the fourth, fifth, sixth position, carbon will be present. In the seventh position, nitrogen will be present. In the eighth position, carbon will be present. And in the ninth position, nitrogen will be present. This is 1, 2, 3, 4, 5, 6, 7, 8 and 9. So, we have to, according to the question, we have to understand what are the amino acids responsible for the formation of this purines, which is having a double ring structure. So, in the first position, nitrogen is present and it is made up of the amino acid aspartate. Okay, and in the second position carbon, and in the eighth position of the carbon, they are made from the amino acid which is formate. The second and the eighth is made up of formate. Next comes the third and the ninth position are having the nitrogen atom and they are made up of glutamine amide. Okay, now next comes the sixth carbon. The 6 carbons are made up of carbon dioxide or bicarbonate. The remaining 3, that is the 7th nitrogen, 5th carbon and the 4th carbon, are made up of glycine. Okay, these 3 are made up of glycine. So, the first nitrogen is made up of aspartate, second carbon is made up of formate, third and ninth nitrogen is made up of glutamine amide, eighth carbon is made up of formate, fourth carbon, fifth carbon and seventh nitrogen is made up of glycine, and the sixth carbon is made up of bicarbonate. So, when we write about the sequence, here it is NCN, CCC, NC, and in this way we will write down it. Now, when we talk about the purines, another thing we will discuss, that is about the adenine and guanine. In which position the NH3 group or the O group is present, and what is the structure of adenine and guanine. See, here we will discuss about adenine and guanine. When we are discussing about adenine, in the sixth carbon, here in this position, one NH3 group will be present. In the sixth position, one NH3 group will be present. And when we talk about guanine, in the second position, NH3 will be present. And in the sixth position, double bond O will be present. See here in the second position, in this position, NH3 will be present in case of a guanine. And in the sixth position, double bond O will be present. Okay, in case of adenine, in the 6th position, NH3 group will be present. Okay, now we will discuss about the pyrimidines. When we discuss about the pyrimidines, it is not a double ring structure, it is having only one ring. Only one ring is present in case of pyrimidines. Okay. Now we will number it. The numbering will start from this side. 1, 2, 3, 4, 5 and 6. Now we will see where the nitrogen and the carbon is present. From this third part, we can write as NC, NCC. Here N, next comes a C, N, then C, C, C is present. Now when we talk about the amino acids, in the third nitrogen, it is made up of glutamine. And the second carbon is made up of carbon dioxide via carbamyl phosphate. And the rest are made up of, and these all are made up of aspartate. Okay, this is all about the pyrimidines. And when we talk about the pyrimidines, that is cytosine, uracil and thymine. In case of cytosine, in the second position double bond O is present. In the fourth position NH2 group is present, which is of single bond. When we are talking about uracil, in the second position double bond O is present, and in fourth position double bond O is present. Now when we talk about thymine, this is same as uracil, only one CH3 group is added in the fifth position. In the fifth position, one CH3 group is added in case of thymine. Now let us see the question. In the biosynthesis of purines, this is a structure of purines. We have to identify which nitrogen is from which amino acid. So in the first option, it has been given that all nitrogen atoms C4 and C5 from aspartic acid. No, we have not discussed about this point, so it is a wrong option. Next comes N1 from aspartic acid. Yes, we have discussed that N1 is for aspartic acid in case of purines. In case of purines, this N1 is from aspartic acid. So in the second option, we are having that N1 is from aspartic acid. In the third option also, we are having that N1 is from aspartic acid. But the fourth option is not having aspartic acid. It has been given that N1 is from glutamine. So we can eliminate this option. So there are second and third option which can be correct. Now, here in the second option, it has been given that N3 and N9 are from glutamine side chains. Okay. In the third option, it has been given that N3 is from glutamine side chain and N9 from N-attached to C alpha of glutamine. In this, we have discussed that third nitrogen and ninth nitrogen is from glutamine amide. So, our second option is the correct option because here we have N3 and N9 is from glutamine side chain and N7, C4 and C5. See here we have N7, C5 and C4. They are made up of the amino acid glycine. It has been given that it is made up of glycine in the second option. So second option is the correct option. Now let us see the second question. The second question is also asking about the same thing. Precursor of the atoms in the purine cytoskeleton are. This is a precursor of the cytoskeleton. We have to identify which amino acid is from which nitrogen or from which carbon. Okay. Let us see the first option. In the first option, it has been given that N1 is from aspartate. Okay, so in the three options, first, second and third, we have that N1 is from aspartate. But in the fourth option, we have that N1 is from glutamine. So we can eliminate the fourth option from here. Now, let us see the first option. C2 and C8 are formate. Yes, we have discussed that C2 and C8 is from formate. So our second option is incorrect because it has been given that C2 and C8 are from citrate, but the correct answer is formate. So in the first or the third option can be correct. Now, in the first option, it has been given that N3 and N9 are guanine and C4, C5 and N7 are glycine. And in the third option, it has been given that N3 and N9 are for amide nitrogen, which is incorrect. And the first option, which is N3 and N9 are from guanidine and C4, C5, N7 is glycine, and 6 carbon dioxide, C6 is for carbon dioxide. Let us see this. Here we have C6 is from carbon dioxide. Bicarbonate can be written as carbon dioxide. Okay, so the first option is correct. Here it has been given that C4, C5 and N7 is from glycine. Here we are having that C4, C5 and N7 is for glycine. So our first option is correct for this question. So these are the two questions which we have discussed to this concept, and from this concept only one statement can also be given, and it has been given in CSIR life science questions. So these two charts which we have, two slides which we have drawn is very important. From this we can solve many questions. So this is all about today's lecture, and thank you for watching this video till the end. And if you like this video, please do like, share and subscribe to this channel.